Students, this is chapter number 4, plant nutrition, and in today's lesson we have to see the transverse section or TS of the lamina or leaf. We call it the internal structure of lamina or leaf. Then we'll see the structure and function of guard cells, though we know a little about it, but let's discuss it in detail. Then we will see how carbon dioxide, water and mineral ions or mineral salts reach the leaf. If you know that leaves are the sites of photosynthesis, and for that they should have the raw material or the reactants for photosynthesis. In our previous lecture we got to know that carbon dioxide and water are the raw material or reactants for photosynthesis, and now we have to see how do they reach the leaf. Then we will see the importance of magnesium and nitrate ions in plants. Let's first discuss the internal structure of leaf. This is the diagram. Don't get panicked or get disturbed by the picture. It's very easy. The outermost layer, which is made up of single layer shown in green color, is called upper epidermis. This is the single layered cells which are deprived of chloroplast. That means that they do not have chloroplast and they cannot conduct photosynthesis. So what do? They are transparent, so focuses the sunlight on the surface of the leaf. They help to focus the sunlight, plus they also protect the inner parts of the leaves from mechanical injury, since they are the outermost layer. And remember students, above it is a waxy layer called cuticle, which is shown in gray color. This cuticle is a waxy substance. It's non-cellular, means it is not made up of cells. What does it do? It prevents the excessive loss of water and also focuses the light into the cells which are lying below them. Upper epidermis, like I told you, is not containing chloroplast and it protects the lower regions of the leaf. Now come to the other cells, which are written as palisade mesophyll cells and spongy mesophyll cells. Both of them are together called mesophyll cells, and their specialty is that they contain a lot of chloroplast. So these are the sites of photosynthesis. Here photosynthesis takes place because of the fact that palisade mesophyll cells contain the maximum number of chloroplast, and then spongy mesophyll cells do contain chloroplast as well. And as you can see that the spongy mesophyll cells also have air spaces between them, which are called as intercellular spaces or simply air spaces. These are lined with thin layer of moisture, the mesophyll cells. All the mesophyll cells, whether they are palisade or mesophyll, spongy mesophyll cells, both of them are lined with thin layer of moisture. Then in the center you can see there are many shapes of cells that you can see. These one colored as in purple color are called xylem. Xylem are the tubular system which is responsible for the transportation of water and dissolved mineral salts. Below them are the other cells called phloem. Phloem is also a tubular structures. These structures are responsible for transportation of sucrose and amino acid, which is made by the plant. Together xylem and phloem are called vascular bundles. I am repeating, xylem and phloem together are called vascular bundles. Spongy mesophyll cells and palisade mesophyll cells are the two type of mesophyll cells, and their specialty is to conduct or to let photosynthesis happen in them, since they contain a lot of chloroplast. Then on the lower side you can see a similar layer as we saw in the upper part. This is called lower epidermis cell. These cells are single layered but they have a adaptation that some of the cells are bean shaped, which are called guard cells, and these guard cells are actually responsible for opening and closing of stomata, which is a pore. Now what's the function of this pore or stomata? Stomata has a very crucial role to play. It allows gaseous exchange, carbon dioxide into the leaf and oxygen out of the leaf. Cuticle is the waxy layer. Like I told you, it protects the leaf, prevents excessive loss of water, and focuses the light onto the mesophyll layer. While upper and lower epidermal cells contain no chloroplasts, they only protect the inner regions of leaf and also focuses the light too. Mesophyll layer, both palisade and spongy, are the main site of photosynthesis since they contain chloroplast. Stomata are the openings bounded by guard cells, and stomata are present abundantly in the lower epidermis compared to the upper one. Guard cells are bean shaped and contain chloroplast. So now we know that in the structure of leaf, 3 sites are there where photosynthesis can take place. First is palisade mesophyll cells, then are spongy mesophyll cells, and then the guard cells. Guard cells are different than the other lower epidermal cells in a way that they differ in shape and as well as in their function. Shape, if you can see in the diagram, the guard cells are bean shaped, unlike the lower epidermal cells which are irregular in shape, plus they contain chloroplasts which are not present in the other cells of the epidermis. They are bean shaped or kidney shaped and contain chloroplast. So what happens during the day? During the day, the guard cells are able to conduct photosynthesis, hence they can produce sugar or glucose. When glucose is produced in them, the water potential will start to drop. Also remember, the potassium ions in the guard cells are higher in concentration compared to other epidermal cells. So what will happen if I will keep on increasing the amount of glucose and potassium ion in the guard cells? The water potential will get lowered, it will get less. So water from the adjacent cells or from the other epidermal cells will start to move into the guard cells by osmosis, leading to turgidity of the guard cells, causing the stomata to open. So this will happen during the daytime, because during the daytime glucose can be formed by photosynthesis, because photosynthesis happens only when the light is present. So guard cells are able to get turgid only when water potential would get lower, and to lower that you need to provide it with solutes. A lot of solute is needed. So what is acting as solute here? It's the glucose which is formed because of photosynthesis, and potassium ions which are already present in the guard cells in higher amount compared to the other lower epidermal cells. So you see, in the daytime the stomata is usually open because guard cells are turgid during the daytime. On the other hand, at night what happens is that sugar is used up by the plant. Water leaves the guard cell, which loses their turgidity and becomes flaccid. As a result, stomata closes, reducing the rate of transpiration. So at night, the guard cells are unable to do photosynthesis because light is not there, sugar is not formed, and whatever sugar is being formed would be used by the cells. So now the water potential is higher compared to the epidermal cells, so water would lose or would go out from the guard cells, leading to closure of the stomata, because now the guard cells have lost their turgidity. Always remember that stomata are open when guard cells are turgid, and stomata are closed when guard cells lose their turgidity. And what would be the effect overall effect on the plant. Here in the diagram you can see that when the guard cell, when the plant is turgid, when the plant cells are turgid, the plant would maintain its upright condition. On the other hand, if the plant cell would lose water, the plant would become flaccid or will wilt. This is the animation to help you understand the opening and closing of stomata, which is controlled by actually the water potential in the guard cells. Whenever the guard cells will be turgid, stomata would be open, and whenever the guard cell will lose water, the guard cells losing their turgidity would result into closure of the stomata. Now let's see how carbon dioxide gets into the leaf. This is the function of stomata, the gaseous exchange. What gases are involved? Carbon dioxide and oxygen. Carbon dioxide needs to go into the leaf for photosynthesis. Oxygen, which is made from photosynthesis, will be liberated and would come out into the atmosphere through the stomata. In addition to these gases, water also leaves the plant through stomata by a process called transpiration. So what processes or what stages happens when carbon dioxide has to enter the leaf? In daylight, photosynthesis occurs. Carbon dioxide rapidly used up in the leaf leads to lower concentration of carbon dioxide in the leaf compared to atmospheric air. This will create a difference of concentration of carbon dioxide. I mean to say that whenever the photosynthesis is happening, inside the leaf the concentration of carbon dioxide will decrease compared to the outside. So carbon dioxide from the external environment would get into the leaf by simple diffusion. Carbon dioxide diffuses from the external environment through the stomata into the air spaces in the leaf. Then it gets dissolved into the moisture lining the mesophyll cells. Carbon dioxide would get into the mesophyll cells only in dissolved form. Always remember that gases are not taken up by the cells in gaseous form. They must be dissolved in moisture or in water to be absorbed. This is the diagram to help you understand the entry of carbon dioxide into the leaf. This is the same structure of the leaf. Lower epidermis, then we have spongy mesophyll cells, we have palisade cells and upper epidermis. So if you can see here, carbon dioxide in the environment is going into the leaf. Why it is going into the leaf? Because of diffusion gradient. Diffusion gradient means that the concentration of carbon dioxide is different in external environment and in the internal environment of the leaf. How they are different? Inside the leaf the photosynthesis is happening, so carbon dioxide concentration inside the leaf is less compared to the outside. So we know that gases move from the area of their higher concentration to the area of their low concentration. So carbon dioxide would move from external environment into the leaf by simple diffusion. First step would be entry of carbon dioxide into the intercellular spaces or air spaces by diffusion gradient. Second point would elaborate the presence of carbon dioxide in the air spaces. Third step would be the dissolving of the carbon dioxide into the moisture lining around the mesophyll cell. This blue dot here is indicating you the presence of moisture around the mesophyll cells. Obviously I haven't drawn all the moisture in all the mesophyll cells, but remember they are everywhere. All the mesophyll cells, all the palisade and spongy cells, they are lined with moisture lining into which carbon dioxide will get dissolved and would enter into the mesophyll cells. Now when the carbon dioxide is inside the mesophyll cell, what would happen? photosynthesis will take place because carbon dioxide is there now, and now plant can use it to make glucose. Now let's see the water entry and mineral salt. This is a topic that we have already discussed in chapter number 1. Water enters through osmosis because of the water potential gradient. If you remember from chapter 1, water outside the soil is greater compared to root hair cell, so it would move from the soil into the root hair cell by simple osmosis. And now once the water is inside the root hair cell, it would be more in quantity or amount, or water potential would be higher here in the root hair cell compared to adjacent cells. So water would keep moving ahead until it will reach xylem vessel. In the next chapter, we will read how water moves into the xylem vessel. But for now, remember that this xylem vessel and phloem are present in the veins of the leaves, and water actually enters from root hair cells, goes into the adjacent cells by osmosis. Since water potential gradient exists here, it reaches xylem vessel, after which water would move into the xylem vessel. The vascular bundles means phloem and xylem, are present in the veins in the leaf. Xylem brings water and dissolved mineral salt to the leaf from the soil via roots. Once out of the veins, these raw materials diffuse from cell to cell. Most of the minerals ions are absorbed by root hair cell by active transport. This is something we have read in chapter 2, that mineral ions are absorbed by root hair cell by active transport. What minerals are involved? The major ones in your syllabus are magnesium and nitrate ions. We have read their detail before, but let's revise them. Magnesium is used for the formation of chlorophyll, which is a green pigment in chloroplast. Chlorophyll actually absorbs the sunlight and converts sunlight into chemical energy, which is used in the process of photosynthesis. Deficiency of magnesium ions would lead to chlorosis, which is yellowing of the leaves and reduced rate of photosynthesis. Nitrate ions, on the other hand, combine with glucose, which is formed during photosynthesis, to form amino acids. These amino acids are the building block for proteins. Proteins are required by the plant for the formation of newer cells or newer protoplasm during their growth, and they also need protein for repair of the worn out parts. Proteins can be stored in plants too. Deficiency of nitrate ions causes stunted or restricted growth of the plants and also causes yellowing of the leaves. Now, what would happen if the plant is lacking magnesium and nitrate ion? Same thing that we discussed in previous slide. Magnesium ion deficiency would lead to reduced chlorophyll formation, yellowing of the leaves, low rate of photosynthesis, low formation of glucose, less respiration would happen since there is less glucose. Less respiration means less energy released, leading to less active transport of ions. On the other hand, nitrate ion deficiency lead to less formation of amino acid and proteins. Less protein means formation of less protoplasm, or fewer newer cells would produce. It would lead to stunted, restricted and poor growth of the plants, and also would lead to yellowing of the leaves.